أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم هل أتى على الإنسان حين من الدهر لم يكن شيئا مذكورا إنا خلقنا الإنسان من نطفة أمشاج نبتليه فجعلناه سميعا بصيرا صدق الله العلي العظيم. Students, I started the discussion of Mendel's law of inheritance in the classroom. I will continue that and you will listen some more information about these two laws. So we are here to discuss the Mendel's law. We were discussing these things in the class. What was the genotype? The genes exist in the form of the pair, and when the genes will combine with each other, they will be segregated during the formation of the zygote in such a way that each gamete will receive the single gene, and in the law of segregation the mathematical ratio was 3:1. 3:1. This was the mathematical representation of the law of segregation. Genes exist in the form of the pair capital R, round phenotype, small r phenotype is wrinkled. The gamete will be shown in the circle capital R, small r, and the capital R, small r will be F1 generation, and F1 generation will be called as monohybrid. It will be heterozygous. One gene will be dominant, one gene will be recessive. Then what will happen? We will self-cross F1 generation. Self-cross mean F1 generation is self-cross capital R, small r, multiply by capital R, small r. These are the generation. Phenotype is round because capital R control the round gene phenotype and small r wrinkled. So recessive gene will not appear character. Dominant gene will appear character. Capital R will appear character. Small r will not appear character. So during exactly the definition of law of segregation, the genes exist in the form of the pair. During segregation of the gene, each gene of the pair will be segregated in such a way that each gamete will receive the single gene. So these are the two gametes. Now this is the Punnett square which is used to ace the calculations. The gamete of the one individual is written here. The gamete of the one individual here, capital R, small r. Gamete of the other individual, other poison plant is here. Now you will combine capital R will go with capital R, capital R, capital R, capital R, small r, capital R, small r, capital R, small r, capital R, small r, small r, small r. This is round, this is round, this is round, this is wrinkled. So 3:1 is achieved. That is the mathematical result of law of segregation. Law of independent assortment. This is interesting. Law of independent assortment is interesting. In the law of independent assortment, the Mendel wanted to know that if more than one gene pair will be taken in the crossings, so what will happen? More than one gene pair will be taken, so what will happen? More than one gene pair will be taken, each gene pair will be independent of the other. I will prove by mathematically by Punnett square, because 9:3:1 is achieved in case of Mendel's law of independent assortment. In case of the Mendel's law of independent assortment. Look here, I will tell you. I told in classroom that two gene pair we take simultaneously, and capital R, capital Y, capital Y, capital R, capital R, capital Y, capital Y. What will be the phenotype? Round yellow. I will write here. I will make, just hold on. I will make the crossing, and I will show you that how these crossings will help in the identification of the independent. Look here, capital R. I should drop this one. Is yes. Look here now. This is very easy now for you. Capital R, capital Y, capital Y. So here one crossing is missed. One crossing I will make, then I will explain from this data. Look here, capital R, capital Y, capital Y. How many gene pairs? We took here? 2. Shabash. and now another. This is the one parent. Second parent I took small r, small r, small y, small y. So it means that capital R, capital R, capital Y was one parent, and small r, small r, small y was one parent. Look here, these were the parental generation. These were the P1 generation true breeders because both were homozygous. Independent assortment law states when both than one pair, more than one pair of the gene took in cross. Normally in our body, one gene pair is not coming from the parents. Many gene pairs simultaneously come. That is the true method to assess the genes. So when what will happen, one family member will not affect other family member. Mean capital R is independent of capital Y, small r is independent of small y. So this is the information I will verify by the crossing. Capital R, capital R, capital Y genotype. Phenotype round yellow, small r, small y, wrinkled green. This is the phenotype. Genotype small r, small y. Now the gametes will be formed. You know genes exist in the form of the pair, and during segregation, segregation will also be valid here because genes becomes half. So capital R, these are two R. One R will come in the gamete because chromosomes reduce in the gamete, genes reduce in the gamete. And capital Y, these are two normal genes. In the gamete they will be reduced. This is the problem. Two different friends are in one car. Capital R belong to one family and capital Y belong to another family. This is the problem. That's the Mendel wanted to know, that capital R will be independent of capital Y or will learn some lesson from capital Y when they will be both together because they both belong to different family. Similarly here in this case, small r, small y, these are different family. Small r, small y. So they will be for some time in the gamete, which is a carrier, will be together. So we will see the results will be affected or not. We will see that. So now they will both combine because in the F1 generation, similar gene will go to similar gene. Capital R will go with small r, capital R will go with the small r because capital R is the allele of small r, capital Y is the allele of the small y. They both will combine and that generation will be called as F1 generation. So F1 generation will be achieved. How the F1 generation will be achieved? Capital R, small r, capital Y, small y. This is the F1 generation, and phenotype will be round yellow. That will be the phenotype. Now in F2 generation, that is very interesting because that is the point I want your attention, because if you will learn the law here, so definitely you will not see much difference in second year. If you listen the second year lecture of genetics which I recorded, that is exactly the same. So my sons, if you will understand the concept here, you will not feel difficulty in senior classes. Capital R, small r, capital Y, small y. This is F1 generation. It is also called dihybrid. You know dihybrid. So dihybrid multiply by dihybrid. If to get F2 generation. Now this is the point which I just wanted to tell. Then you will understand the independent assortment, key of independent assortment of the genes. Capital R. This capital R, I should magnify so that you form the gamete. Yes, this is easier one. This capital R can go with capital Y as well and can go with the small y as well. So chances to go with capital Y is 50%. The chances to go with small y is 50% because capital R is not depending upon capital Y. That is a different family. So capital R is not friend of capital Y. Capital R says I can go with capital Y, I can go with the small y. This is the point. So one family is independent of another family. Capital R, small r, one family. controlling round color, round shape and wrinkled shape, and capital Y, small y controlling yellow color of the seed and green color of the seed. And they both families, capital R, small r, are independent of capital Y and small y. So capital Y says, small r says, I can go with capital Y as well, I can go with the small y as well. 50% chance. So that is the reason variation comes in our body. In the animals, some characters are parental and some characters are non-parental. I will confirm that how some characters are parental and some characters are non-parental. This 50% ratio, this 50% chance, this independent assortment of the genes, this independent decisions of the one family and independent decisions of another family create the variation which I will tell you, because some characters in your body belongs to your father and mother and some characters do not belong to father and mother. Reason is only one, because the genes are independently assorted independently assorted. I will tell you how the genes are independently assorted. These are the gametes of the one individual, these are the gametes of the another plant, another male and female. So for the Punnett scale you will use this Punnett scale. Gametes of the one animal, gametes of the one plant, Pisum stivum, male should be written at one side. Similar genes are there, similar gametes are there. Here also the similar gametes are found. 4 similar gametes are in the left side. So now they can combine with their family members, because they will be in different mode in the gamete only. In the gamete only 2 different family members were there, and Mendel was scared might be they affect each other. They don't affect. That's why this law is called independent assortment of the genes. Now in offspring of F2 generation, this is F2 generation, and in the F2 generation what will happen, that capital R will go with capital R and capital Y will go small y. So it is very easy to make, you can make it very easy. Anyone I select anyone. Look here, I am making this one and this one. Okay, I am making this one and this one. Okay, look, capital R go with own family small r, and capital Y, just capital letter you will write early and capital small letter you will write later if that comes. Now after that, remarkable results were there. What were the results? 9:3:3:1. What is the meaning of this? Meaning of this is that some phenotypes were parental, mean round yellow, this is round yellow were 9, and some parental, characters, some phenotypes were non-parental phenotypes were achieved. What were the non-parental phenotypes were there? I tell you. For example, this one, round green, and this is round yellow, this is parental because it is present in the parents. I show the parents once again you. These are the parental. Round yellow is the parent and wrinkled green is the parent. So what is the mathematical ratio? Mathematical ratio is 9:3:3 and ratio 1. Okay, look here, what are parental? 1 is parental, 9 is parental, 3 is non-parental, 3 is non-parental. Why non-parental generations are produced? Non-parental generations are produced because capital R had the chance to go with capital Y. When it will go with capital Y, it will be look like parent. Reason, because in the parent capital R is with capital Y. When capital R go with small y, 50% chance that capital R can go with the small y, that is 50% chance, and that is the 50% chance that the capital R can go with capital Y. Capital R can go with the small y, 50% chance. If capital R go with capital Y, this is parental generation, parental character, because in the parents capital R is with capital Y. If capital R go with small y, this is non-parental, new character will be found, new phenotype will form. So 9:3:3:1 mean two two generations are parental and two generations are non-parental. In the parent, capital R was with capital Y. In the parental, small r with the small y. Therefore, if the capital R goes with the small y, the new parental character will be in the Punnett square as it is achieved. You can see capital R goes with small y, this one I can show you. So this is non-parental character. Similarly, if the small r, where is the parental generation? If the small r with the small y, so this is father and mother character. Where is the father and mother character here? This is the father-mother character in the offsprings. When small r goes with capital Y, it means this is not parental character. This is the non-parental character. Small r goes because small r goes with small y. So by this way, four generations are produced. Two of them are parental and two of them non-parental. Parental mean no variation, and this mean variation took place. Change in character, change in genetic material is called as variation. So these were informations about Mendel's law of segregation and Mendel's law of independent assortment.